Okay, so the question states that squid is an animal that is eaten in many parts of the world. The body of the squid is used as meat. It is used as meat. Why? Because it contains a lot of protein. The photograph shows some squid meat. The meat from the squid is quite tough and is often treated with enzymes to break the protein down. Some fruits contain the enzymes that can break down the protein. Pineapples contain the enzyme bromelain, and unripe papayas contain the enzyme papain. A study compared the effects of two different concentrations of these two enzymes on the meat of the squid. तो ये हमें क्या बता रहे हैं कि जो मीट ऑफ द स्क्विड है इसके ऊपर हमने दो एंजाइम्स को टेस्ट किया है, एक है ब्रोमिलिन और एक है पपेन. ब्रोमिलिन की हमने दो कंसनट्रेशंस बनाई हैं, पपेन की भी हमने दो कंसनट्रेशंस बनाई हैं. ठीक है, तो in total we have four different concentrations that were enzymatic concentrations that we're going to test on this particular meat कि ये प्रोटीन को कितना ज़्यादा ब्रेक डाउन करता है. After understanding the question, we move on to the next information that they've given us. The part of the squid that cannot be eaten was removed and the remaining meat was cut into equal sized pieces. This is important कि meat को equal sized pieces में काटा गया है. Next, each piece of meat was placed into a beaker containing either a solution of bromelain or a solution of papain or a control solution. ठीक है, एट अ कांस्टेंट पीएच दैट इज पीएच 7. तो उन्होंने बता दिया वन ऑफ द वेरिएबल दैट दे केप्ट कांस्टेंट इज द पीएच. सेकंड, दे टोल्ड अस दैट व्हाट दे आर टेस्टिंग थ्री डिफरेंट things. They're testing two different enzymes and the third solution is a control solution जिसके अंदर एंजाइम नहीं होगा या अगर एंजाइम है तो वो एंजाइम अपनी एक्टिविटी परफॉर्म नहीं कर सकता. Step 3 is that the beaker was incubated to allow the enzyme to act on the meat, the pH and the soluble protein. आखिर में टेस्ट क्या कर रहे हैं, pH टेस्ट करेंगे, soluble protein, कंटेंट ऑफ ईच ऑफ द सोल्यूशन वॉज मेजर्ड. पीएच देखेंगे कि पीएच ज़्यादा हुआ है 7 से या कम हुआ है. प्रोटीन देखेंगे कि एंजाइम ने कितना प्रोटीन ब्रेक डाउन किया है. So the first question they are asking that explain why in step 1 it is important to cut the squid into equal sized pieces. अब स्क्विड को हमने इक्वल साइज पीसेस में क्यों किया है कट, ताकि हमारे पास सरफेस एरिया हर एक पीस का सेम रहे. ठीक है, जब हमारे पास सरफेस एरिया सेम रहेगा तो उससे क्या होगा, उससे ये होगा कि जो हमारे पास एंजाइम है दैट वुड हैव equal number of substrate to act on, and when that happens that produces a valid result. So we cut this meat into equal size pieces so that the surface area is same. When the surface area is same, then the enzyme has equal amount of substrate to act on, and therefore this produces a valid result for the experiment. That is how you're going to write your answer. The second thing that they say is that name a suitable control solution that could be used in step 2. अब whenever we talk about control उसका मतलब ये होता है जो चीज़ हम टेस्ट कर रहे हैं वो चीज़ उसके अंदर नहीं होगी. ठीक है, तो कंट्रोल हम इसके अंदर क्या कर सकते हैं, हम टेस्ट किसको करें, हम एंजाइम की एफिशिएंसी को टेस्ट करें कि कौन सा एंजाइम कितनी पीएच चेंज देता है, कितना प्रोटीन कंसंट्रेशन इंक्रीज करता है. ठीक है, तो अब एंजाइम को अगर हमने रिप्लेस करना है हम उसको कैसे रिप्लेस कर सकते हैं, या तो हम उस सॉल्यूशन में वाटर डाल सकते हैं. ठीक है, दूसरी चीज़ हम क्या कर सकते हैं, हम लोग एंजाइम को डीनेचर कर सकते हैं. ठीक है, आइदर ऑफ द टू थिंग्स, क्योंकि पानी भी एक्ट नहीं करेगा सबस्ट्रेट के साथ, ना डीनेचर्ड एंजाइम एक्ट करेगा सबस्ट्रेट के साथ. सो आइदर ऑफ द टू थिंग्स वी कैन यूज एज अ कंट्रोल इन दैट स्टडी. द नेक्स्ट थिंग इज दैट स्टेट द इम्पॉर्टेंस ऑफ यूजिंग अ कंट्रोल इन द स्टडी. कंट्रोल हम क्यों यूज़ कर रहे हैं, कंट्रोल हम इस वजह से यूज़ कर रहे हैं सो दैट any effects can be attributed to the enzymes and not the solvent that is present there. ठीक है, यूजिंग अ कंट्रोल गिव्स अस अ बेसलाइन कि जो भी इस पर्टिकुलर रिएक्शन में अगर तीन हमारे पास एंजाइम्स हैं, ये ब्रोमिलीन है, ये हमारे पास पपेन है और ये हमारे पास कंट्रोल है, इसके अंदर जो भी चेंज इन कंसंट्रेशन हुआ है वो चेंज इन कंसंट्रेशन कंट्रोल में नहीं होगा, और ये हमें बताएगा कि इन दोनों में जो भी चेंज आया है वो इसी वजह से आया है क्योंकि इनके अंदर ये दोनों एंजाइमेटिक एक्टिविटीज़ कर रहे थे, ये दोनों एंजाइम्स. कंट्रोल में देयर वुड नॉट बी दैट मच ऑफ अ चेंज. Next question that they ask is, Give two variables that need to be controlled in step 3. They already told about two variables that they did control. Those two variables were pH. They kept the pH constant at pH 7. Second, they use equal sizes of meat. Now, apart from these two, you need to write other two. Okay? So, the other two could be temperature. Temperature needs to be kept constant while incubation is happening. And the second thing that they need to keep constant is that the duration of time needs to be कांस्टेंट. जितनी देर के लिए सबस्ट्रेट को एंजाइम के साथ रिएक्ट करने की इजाज़त दी गई है, दैट शुड बी इक्वल फॉर ऑल ऑफ द सैम्पल्स. नेक्स्ट वी आर गोइंग टू टॉक अबाउट दैट द ग्राफ शोज द पीएच ऑफ द सॉल्यूशन दैट वाज मेजर्ड एट स्टेप 4. ठीक है, अब यहाँ पे हमारे पास आ जाता है कंसंट्रेशन ऑफ एंजाइम. ठीक है, यहाँ पे आ गया पीएच कितना चेंज हुआ है. हमारे पास दिस इज द कंट्रोल रिमेन्स एट पीएच 6.6, 6. This remains over here. ठीक है. Second, we're going to look at bromelain and we are going to look at papain. ये it is the lower concentration. This is the higher concentration. How much did the pH change? We can see that the pH changed more in that of bromelain. bromelain ने ज़्यादा pH change किया है, यानी कि ज़्यादा activity perform की है. Second चीज़ we can see कि 0.004 की जो concentration है उसमें ज़्यादा activity हुई है, क्योंकि उसमें pH की change as compared to the control ज़्यादा थी. ठीक है. Now this we have just understood कि क्या चीज़ हो रही है. Next we are going to look at the questions. The first question states that 0.001% enzyme solution was prepared by diluting the 0.004% enzyme solution. Calculate the volume of the 0.004% enzyme solution needed to make 50 centimeter cube of 0.001% enzyme solution. यानी कि इसमें हो क्या रहा है? In order to understand this, you need to understand that this is a dilution problem. We are talking about dilutions over here. Whenever we're talking about dilution, this means that we're adding more volume of the stock. We're adding more volume of, let's say, water. But what is remaining constant through it all? The thing that is remaining constant through it all is the amount of solute remains constant. Only the volume is changing, right? So if we look over here, the formula we're going to use is concentration 1 into volume 1 equals to concentration 2 into volume 2. Okay. So the initial concentration is 0.004. However, we do not have the volume for it. The resultant concentration is 0.001 and the volume for it is 50 centimeter cube. If we simplify this, then what we can do is this: V1 equals to 0.001 into 50 divided by 0.004, and this would give us an answer of 12.5 centimeter cube of the volume. Okay, so since this was a dilution problem, therefore we use this particular equation. Next we're going to see, they are saying that state two conclusions that can be made from the data that has been shown in the graph. So we have already discussed that as well when we were understanding the question. The description should include, the first thing that needs to be there is that both enzymes decrease the pH. So what we're looking at, there is from 7, there's not 7, but if we say that from 7, there is no increase in pH. Both of the enzymes are decreasing the pH. They are making it more acidic. So both the enzymes decrease the pH, that is the first point. And they decrease the pH more than the control. And at a higher concentration, the decrease in pH was more. Then another thing, that the decrease in pH in the solution of bromelain was more than that of the in papain. Okay, the second thing that you need to know, or that you need to write, is this: that bromelain is more effective at breaking down the protein than papain. Since the change in pH was more in bromelain, therefore it is more effective in breaking down the protein. And lastly, what you can write is this, that at higher concentration, both enzymes are more effective at breaking the protein down than that at the lower concentration. You can mention 0.004 and you can give your reference values from here as well to support your answer. Okay. Next, we're going to talk about this. They're saying that describe a method for estimating the soluble protein concentration in the beakers in step 4. So basically, they're asking us to design an experiment where we're going to find the protein concentration. Okay, so obviously we're going to, what test we're going to do, that is the first point that you're going to write. We're going to use the Biorets reagent in this test because we're testing the protein concentration. Okay, or we're going to do the Biorets test. Next, what you need to write is this, that you're going to remove the meat from the solutions that they were in, and you're going to take the sample of the solution only. So you need to filter the solutions, remove the meat, and then take the sample of the solution. When you're taking the sample of the solution, you need to ensure that you're taking equal volumes of the sample for testing. Okay, and you're going to use equal volume of Biorets reagent as well. Lastly, you're going to tell that how you're going to calibrate, how you're going to find the concentrations. So you're going to make a calibration curve, and the concentrations that you're going to find, by comparing the color to standard colors. You're going to compare the colors to standard colors. Concentration, and you're going to use, what you're going to use, you're going to use a calorimeter for that. So using a calorimeter, compare the color to the color standards, and then you're going to make a calibration curve. And this is how you're going to do the Biorets reagent test. So we're done with the question 1 over here. Okay. Okay. So the second question says that little tunny is a fish found in the Atlantic Ocean. The photograph shows a little tunny. Calculate the actual length of the fish between x and y. Give your answers in meters. So they've given you the magnification. Now in the paper you'll require to draw a straight line across the fish and then measure that particular straight line. Okay, over here we don't have that straight line, so let's say if the answer gives us 9.8 centimeters. Okay, so this means that the image is 9.8 centimeters. So firstly, since we have to give the answers in meters, हम उसको meters में convert कर लेंगे. We know that 1 meter equals to 100 centimeters. So 9.8 centimeters would be how many meters? It would give us, if we divide it, it would give us an answer of 0.098 meters. Now this is the image. So the formula that we use is IAM. We need to find the actual length. So 0.098 divided by the magnification, that is 0.09. When we divide it, we get the answer 1.08. So we can say that the actual size is 1 meters. Okay. The next thing that they ask us is this, that a student wanted to compare the heart of this fish with the heart of a mammal. So the student planned to cut open the fish and remove its heart to keep the heart in a liquid preservative called formalin. The student made a risk assessment before dissection. The diagram shows part of the safety data sheet about formalin. Formalin is what? In which the heart will be preserved. So what does the safety data sheet says? It says that formalin is harmful. It is harmful by inhalation, it is harmful by contact on skin, and it is harmful if swallowed. It is irritating to eyes, the respiratory system, and the skin. There is limited evidence of this being a carcinogenic effect, meaning that इसके अंदर इतने ज़्यादा chances नहीं है that exposure to this can cause you cancer. It may cause sensitization by skin contact. So what they ask us in the question is this, that the student put the risk assessment into a table. Complete the table to show one hazard for the dissection and one for the preservation of the heart. तो वो ये बता रहे हैं कि आपने हैजर्ड बताना है. जो पहला हैजर्ड आप बताएंगे वो डाइसेक्शन का बताएंगे कि डाइसेक्शन करते हुए क्या चीज़ आपको नुकसान दे सकती है एंड हाउ टू रिड्यूस द रिस्क. ठीक है, तो डाइसेक्शन करते हुए बिकॉज वी आर गोइंग टू यूज अ स्कैल्पल, वी आर गोइंग टू यूज अ नाइफ, तो हैजर्ड इज यूजिंग अ शार्प scalpel. And how can we reduce the risk of it? We can reduce the risk by cutting in a way that is away from our fingers, okay, and or by wearing gloves. Second hazard during dissection could be any sort of allergic reaction, because there are many people who are allergic to seafood. So in that case you're supposed to wear gloves, you're supposed to have anti-allergy with you, okay. Now the second, they said that the second you need to say tell from the preservation. So that means how is formalin hazardous for you, you need to talk about that. So inhalation of formalin is harmful to you. Why? Because that is an irritant for the respiratory tract. Spilling of formalin is harmful to you. Why? Because the skin is sensitive to it. So what you're supposed to do, that you use this in a well ventilated room. Why? Because when you are in a well ventilated room, the fumes are not going to accumulate. That has an exhaust system, okay. Second, you're supposed to wash your hands as soon as you're done with the experiment, and you're supposed to wear your protective gear. So this is how you're supposed to answer this question. Next, they say that the diagram shows the heart of a fish. Compare and contrast the structures of the heart of a fish with that of the heart of a mammal. So if we look at the fish, this has a single circulatory system. It has only two chambers, and it does not have arteries and veins. It has a sinus venosus and it has a bulbous arteriosus. It has one atrium, it has one ventricle, okay. However, atrium walls are thinner as compared to the ventricular walls, which is same as that in mammals. So if we need to compare and contrast, first thing that you can write is this, that talking about the similarities, when you need to compare and contrast, you can first write about the similarities. What are the similarities? The similarities is that both hearts have one atrium and ventricle, okay. They both have atria and ventricle. And the second thing, that the ventricular walls are thicker as compared to the atrial walls. And another thing, that there are valves present as well, okay. So valves are present as well. The difference that you can point out where you're going to compare and contrast, you can say that the fish has a single circulatory system. It does not have four chambers, it only has two chambers. It does not have arteries and veins, it only has sinus venosus and bulbous arteriosus, okay. Then you're going to compare that the mammals have double circulatory system. They have four chambers, they have arteries and they have veins, okay. So this is how you're going to secure your marks over here. Next, if we look at the question, it says that a student suggested that larger the fish, larger will be the heart. Devise a method that could be used to collect and analyze. First you need to collect the data, then you have to analyze the data as well to test this suggestion. So how are you going to do that? So you need to keep in mind, you need to talk about the collection process and then you have to talk about the analysis process. So there are two things that you're going to talk about. So how you're going to do that? You need to say that you're going to use multiple fish samples or different species. At least you can use, let's say, 5. They need to have different sizes. This is essential. Then what you're going to do, you're going to measure the length of each fish. And how are you going to standardize this? You're going to standardize this by saying, let's say that you're going to measure from the tip of nose to the end of tail. This is how you're going to standardize it, right? Then you're going to measure the length of heart. You can standardize that by saying you're going to measure from the bulbous arteriosus to the sinus venosus to standardize it. Okay. Then you're going to say that before measuring, you're going to remove any excess water or parasites that might be present or anything that is present on the fish. Then now this is the collection method and what you're doing. Now coming to the analysis method, what you're going to write is this, that once you've taken the lengths, you're going to plot the graph. How you're going to plot the graph? You're going to plot the graph of the fish size on the x-axis and the heart size on the y-axis, and then you're going to take the ratios of it. And then you're going to correlate these findings. Okay, you're going to compare the ratios. So this is how you can collect and analyze the size of the heart and the size of the fish. Okay. Okay, so over here we're done with the question 2.